So today we're doing microscopy and cell structure. So what we'll probably focus on mostly today is going to be all the different types of microscopes that we use in microbiology, as well as getting into some of the morphology. Why do we call certain bacteria certain names? And so for the majority of what we're going to be looking at in this class is that the microscopes are going to be mostly for bacteria and the funguses and molds and whatnot that we have in eukarya. Viruses are very, very small, right? They're even smaller. I mean, they're incredibly small. Like we pointed out before, that if we had a bacteria the size of this building, a virus would only be about the size of this outlet. And so, we have to have incredibly strong microscopes to see viruses. And so there's only two that we really have that we can see viruses, and those are going to be the electron microscopes. So just keep in mind, most of what we're talking about today is going to be how we look at bacteria. Not going forward. I figured we'd have a problem somewhere. All right. So light microscopes are the ones that you're used to seeing all the time. These are the ones that you have in your classrooms, your laboratories, things like this that you're used to seeing. So they have a magnification of up to 1,000x. So when you're talking about X, what are we talking about? We're talking about the eye. The eye is 1x, so 1,000 times better than what the eye can see. And so they're common for, most microscopy. Everybody has one in their laboratories, if not multiples. The next one up is going to be the electron microscopes. They were discovered in 1931, or first made in 1931. They can magnify up to 100,000. So here you notice it says that it's good for virus particles. That's about the only way we can see a virus. We can't see a virus with the normal microscopes. As long as the microscope has a light on it somewhere, then it's considered a light microscope. All right. So the electron microscopes are using a beam of electrons to build the image. And so that's where it gets a different name. So light microscopes can be as cheap as the ones you go buy at Walmart for a birthday party or something like that for kids. They come around 60 to 100 bucks. Ones we have over in the microscopy lab or the microbiology lab, they're about $2,000 to $3,000. But then some of the more expensive fluorescent microscopes and confocals that we're going to talk about in this chapter go as high as $500,000 or better. So they're all light microscopes. They all have some sort of light source on them somewhere. Third one down there is an atomic force microscope. They have a very limited ability to for like regular use. These are very specialized, and they're going to be designed to actually look at the atomic structure. And so they can actually get down and look at the atoms and the molecules. And so not a lot of people have much use for them, so it's going to be very niched in terms of who has access to them. I've never even seen one before in 20 plus years of doing research. So the principles of light microscopy. What I want you to focus on these chapters is the different microscopes and what their uses are for. Don't focus on the physics behind them, because it gets a little complicated trying to talk about all the different lenses and the way that the lights go and the way that the light travels through the lenses. Focus on the name and what they're used for. So first one, bright field microscopes. These are the most common type. These are the ones that you're used to seeing. Bright field means that when you look in look into the optical, you're going to see white light, right? And that's what we're used to seeing when we look at a microscope. All microscopes have three major concepts in terms of what they do for us. One, obviously, they're magnifying, right? That's usually the first thing we always think about is, why are we even using a microscope in the first place? So we can magnify what we're looking at. The modern microscopes are compound. When we use the word compound, that means that they have two or more lenses in them. So in this case, the ones that you're used to seeing, you have the oculars that you look through, right? Those are the 10x usually. And then the actual objectives that we can actually turn. Those are going to be the second lens. And so that makes them compound, being two or more. So almost all microscopes nowadays are two. However, single lens microscopes, those are going to be things like glasses on your head if you're wearing glasses. It could be magnifying glass if you're using one of those, just a single lens. That would be also a type of microscope. The magnification of a typical student microscope or standard microscope is going to be 10, 40, and 100. And on the objectives and the oculars are always 10. You can go out and you can modify these. They have different ones out there that you can buy. But ultimately, what we do to figure out what the true magnification is we multiply them together. So if you have an ocular of 10 and you're using the 10x objective, then your actual true magnification is 100x. 10 times 10 equals 100. So when we talk about having the bright fields be able to go up to 1000x, that's using the 100x objective multiplied by the 10x ocular, and that becomes the 1000x. That's pretty much where they max out at. So principles of light microscopy, moving on. The other thing that microscopes do for us besides magnification is they add resolution. Resolution is the ability to tell two spots apart. So the closer things are together and the smaller they are, it's harder for our common eye to see that there's actually two things there. So if I was to draw two little dots up here, those of you that are closer would probably see that there's some space between the dots, but back in the back, it'd be a little bit harder to tell. A microscope, if it magnifies, it also has to be able to increase the resolution. The way that you all probably are used to seeing this is if you take a picture of your phone and you really like it, and you try to blow it up in an 8x10, what happens sometimes? You can't, right? It becomes blurry, it becomes pixelated. Why? Because the resolution is not that great. The newer phones, it's not a problem anymore, but if you're older, you remember the original iPhones, the original phones that had the simpler cameras on them, the resolution was terrible on them. So here you can have terrible resolution on a microscope. And if that's the case, then it doesn't matter how high the magnification is. If the resolution is not good, it doesn't matter, right? You're not going to get a good picture. And so it depends on the quality and type of the lens. Maximum resolving power is about 0.2 micrometers. So this U here is actually the Greek U. We use it in science to determine micro. So when we're using the prefix and metric system micro, we always use the Greek U. And so that's what that's referring to there. So remember, a micrometer means that 1 million micrometers fits into a meter stick. So you're talking incredibly small, and then we're only talking about 1/5 of one of those. So it shows you how incredibly good the resolution can be in these microscopes. Now, some ways that we can actually increase the resolution is by trying to increase the amount of light that goes up into the objective. The more light, the better your resolution is going to be, the better your image is going to be. And so some ways that we do that with the microscopes that we have out in the laboratory is that most of the ones that we have in the lab are going to be air objectives. That means there's nothing between the slide and the objective besides air. Well, what happens, as you can see in this image, is that the light sometimes escapes because there's nothing there holding it. There's nothing grabbing it. And so a lot of your light is actually escaping, and I demonstrate this in my freshman labs by shutting the lights off and just turning one microscope on. Everybody in the room can see that light is on. That means there's a lot of light that's escaping. So when we look at the 100x objectives, we actually use what's called an oil immersion objective. What we do is we put a little drop of immersion oil between the slide and the objective, and what that does is it captures more of the light. More of the light is actually captured and forced through the objective and not actually lost out to the sides. And so that actually helps us increase resolution. As our magnification goes up, we also have to bring up our resolution too. So we refer to those as the refractive indices. Every different media has a different refractive index. We do these back in some of our STEM outreach where we have the pencil in a cup of water, and if you look at it just right, it's an optical illusion. It looks like the pencil's actually broken. Well, that's just because of the refractive index of water versus air, and so it shows that there's just a little bit of a difference in the way that it looks. Microscopes are working underneath that same principle. The third thing that microscopes can do for us is they increase contrast. Contrast is going to be the ability to see the cells that we're actually looking at. So it's basically the shadowing or contrasting, essentially, using the same word of the image. So if we have a really bright white light going through and we're looking at something that's transparent, you're not going to see it. It's just going to look all whitewashed when you look into the oculars. So we have to have an ability to change the contrast. On the microscopes themselves, if you remember, they have the iris diaphragm that we can open and close, and it kind of controls the amount of light that comes through. Most of the microscopes also have a dimming on them where we can change the amount of light coming through. But, we have to do more than that. So in microbiology, we're actually going to use a lot of stains. So we're going to talk about a bunch of stains in this chapter. The stains, what they do is they increase contrast, but unfortunately, they kill the micro. So we lose, if it's a precious sample that we're trying to keep alive for research purposes or whatever it might be, we may not want to stain them, because if we do that, we're going to kill them. And that's not a good thing sometimes. So we do have some microscopes that we can utilize that actually we look at live specimen and not have to stain them. So the first set of microscopes we're going through are going to be ones that we can actually look at live specimen. The rest of them we're going to be looking at stained or potentially killed specimens. Here's dark field microscopes. Dark field microscopes direct the light towards the specimen at an angle. So it's actually changing the way the light is hitting the specimen. And it's going to change the way that it actually is going to show up on the image too. The best way to think about a dark field is it flips what used to be light in the dark and what used to be dark in the light. All right, so what we see here is an actual image. And normally, if you remember looking into a microscope, all this background should be white, right? But here in a dark field microscope, it's more of a black color. And now it allows, in this case, this algal, this algae that they're looking at, it allows it to pop. And now we can actually see it in a little bit more detail. And so that's a great thing, for being able to see specimen that maybe it'd be difficult to see underneath the bright field. With the dark field, we can actually kind of get it to pop there and we don't have to kill the specimen. The next one is phase contrast. Phase contrast is referring to light waves. And again, like I said, I don't want you to focus so much on the actual physics, but if you want to understand phase contrast, we have to think about the waves that light travels in. So this is a normal light wave. In a phase contrast, what it's doing is it's changing the phase of that wave. And so now it may go something like this. And sometimes by changing the phase, we actually get better images. And so these are, phase contrast is like an option we can buy on a microscope. So like some of the microscopes we have over in the microbiology lab have the phase contrast option. So they would be normally a bright field, and there's a little slider that you pull out on it, and it changes it to phase contrast. Is it going to work every time to give you a better image? No, but sometimes it does. And so the images that we get makes the cells, as it says here, makes the cells and other denser material appear a little bit brighter. And so again, we can get some more imagery to pop out. We don't have to kill the specimen. And so that becomes important. The last one here is differential interference contrast, or DIC microscopes. And these are going to be very similar to phase contrast, but they're going to have two light beams instead of just one. And those two light beams are going to go out at different angles, and then they're going to come back to the ocular in a way that it almost gives a three-dimensional appearance to the specimen that we're looking at. So in this case, it's really useful. We're looking at some sort of amoeba probably here, and we can actually see the individual vacuoles, and we can almost sense that there's a three-dimensional aspect to them. So all three of these microscope options are great for being able to see the image of what we're trying to look at without actually having to destroy the specimen. But at the end of the day, it really doesn't give us a whole lot of detail. There's not a lot we can do with a specimen that we're trying to keep alive. So the next ones, all these forward, are going to be ones that we actually have to stain. We have to stain, and then we can see them. So this is probably one of the most popular types of microscopes. I think every single microbiology laboratory I've worked in has fluorescent microscopes. And this is going to be talking a little bit about immunology, even though we haven't got the immunology yet. But what they do is that we actually fluorescently tag what we're looking for. And the way that we do that is we tag them with antibodies. Antibodies are very specific in what they're looking for. They will not go and attach to anything besides what they have been designed to go attach to. So our immune system makes antibodies naturally. So when you get sick, why does it take a couple days for you to start feeling a little bit better? Your antibodies have to start building up. Your immune system goes out and starts attacking whatever it is that's making you sick, starts making antibodies against that directly. And then our body then can clear whatever it is that's making us sick. Well, we take advantage of that in science. We, utilizing biotechnology, we basically utilize the idea of antibodies, but we can force antibodies to be produced in a way that they are attaching to what we want. So here, what we're looking at is probably some antibodies that are attached to the outer membrane of these bacteria. And so what we do is we basically will take the bacteria that we're wanting to stain. We stain them with the fluorescent antibody. We wash off any excess antibody because antibodies are incredibly sticky to what they actually want to stick to. So once they find what they're going to stick to, they don't come off. And so what we'll do is then we'll wash it, and then we'll put it underneath the microscope, and it's going to have a laser in there that when the laser hits those fluorescent markers on those antibodies, it makes them glow. And it's actually pretty phenomenal in the way that we can utilize this technology nowadays. Before, when we were doing infectious disease research, like say using a mouse, and we would feed the mouse like whatever we were looking at. In that case, it used to be salmonella when I was working in that lab. We'd have to do a complete necropsy and pull apart the mouse and try to figure out what organs, where that actual bacteria ended up inside the mouse. Now I can actually fluorescently tag for the salmonella, and I could actually see, without even having to go pull every single organ out, we can actually see which organ the bacteria is in. And that makes it a lot brighter. It's a really bright color to it, and it's really easy to see. It's unmistakable once you see a fluorescent image. There are literally thousands and thousands of different antibodies out there nowadays. Back in the 90s and early 2000s, people were developing them in their own labs and then would sell them to companies. And now there's entire libraries of them out there. So this is pretty common. Almost all microbiology labs have a fluorescent microscope in them somewhere. So we'll be seeing a lot of fluorescent images as we move forward. Now, some other microscopes, we got the scanning laser microscopes. They are very good at looking at the interior details of a cell. And we have to kill them. So we're looking at the cell after it's been basically smothered to death with the stain. Imagine if I dumped you in a bunch of paint, which is all a stain is really. I mean, you would suffocate, right? So the same thing's happening to bacteria. Is how they basically end up passing away. Here we're staining them with a fluorescent dye, and then we can actually look at the interior details using these scanning laser microscopes. Confocal are going to be using lasers and also fluorescent tagging. And what we can do with a confocal is it's similar to getting a CAT scan. And so we can actually look at individual slices of the cell, piece by piece by piece. And this is incredibly important for understanding how interactions are happening between, like say, an infectious agent and the cells that it's attacking, or how the immune system is interacting, or in this case, it's just showing us some actin, and then the red here is probably, I can't remember what these images exactly are, but I think the red are the nuclei of the cell, and then we can actually see some of the cytoskeleton there in green. So with the fluorescent microscopes, we can actually use more than just one color too. So in this case, there's two, and some of the newer ones actually go up to multiple colors. Multiphoton is going to allow us for time-lapse images. So if we actually were looking at time-lapse photography type things, penetrates deeper and gives us interior views of something that really thick. So if we're looking at maybe like a biopsy, it's a little bit thicker than just what's like a smear that we can put on a stain or on a slide. This is going to allow us to be able to see that with the multiphoton. So which ones have I seen? I've only seen the confocal microscopes. We got one at Tulane University. My good friend Kirsten is the person that runs it. So all these different microscopes, when we get up to this level, these are incredibly expensive. And usually you have a technician that is their only job. So if you were a research scientist, you would make your slides and you would hand it over to the technician. They would give you a timeline of when they're going to get it back to you, and that's pretty much the job. So you wouldn't actually go and do confocal on your own, right? It's complicated. It takes an entire career's worth of understanding to get the images just right. All right, so we move over to electron microscopes. Again, these are the ones that can go up to 100,000 X, and they're going to use a beam of electrons. So here on the left is showing you the standard light microscope going through the condenser lens, going into the objective lens, seeing the image, coming into the ocular, and then ultimately to the eye. So if you kind of inverted it, probably make more sense. But electron microscopes actually are set up in the way that it is here on the right. Instead of having that light, you have an electron gun, is what they call it. And the electron gun is going to be sending down electrons. It's going to hit the image, and those different electrons are going to bounce off at different angles. And so those different angles that it bounces off on there are going to be different pieces in there that are going to capture those beams, and it's going to basically develop an image on the computer screen. So different way how those beams come off, different angles. It's going to develop that onto the screen or a micrograph, and then you're going to get an image. And so these are incredibly complicated. Usually it takes a good solid 2 or 3 days just to get your slide ready to go. A lot of toxic materials and stuff. So not a lot of fun to be able to work with them, but nowadays what's happening is that they're actually coming out with plug-and-play electron microscopes. Hitachi has one. It's only like $60,000, which sounds only right, but in the grand scheme of things, the old electron microscopes were up towards $1 million. So they're starting to get a little bit better, a little bit easier to use. But when I was going through research, there was no way that I would ever get anywhere close to an electron microscope because I was a bacteriologist, not a virologist, anyway. All right, so there's 2 major types of electron microscopes. There are the transmission and the scanning. The ones that are the most common are the scanning electron microscopes. The scanning allow us to see the exterior of something. The transmission ones allow us to see the interior. So in this case, the scanning here is, well, first of all, that's what an electron microscope looks like. So nothing like your normal image of a microscope, right? It's more of a desktop type thing. It looks more like a computer system than anything. And here's the actual microscope. You put in your image in there, and you actually have to seal it down because it builds a vacuum.
Which one did you say was more common?
More common is the scanning electron microscope. Yep. And so what we can see here, we can actually see a virus molecule right here. So we can actually get down and actually see the virus. In that case, it might be flu there almost. Here, anybody know what that is? A flea, actually. Yeah, close enough. Flea. So we can actually see its eyes. We can see its 2 little, fangs there. We can actually see its, a skeletal structure. If you've ever seen a flea, it's just a little black speck, right? Jumping around. If you have animals. Here you can actually see what it actually looks like up close. And so this is what an electron microscope does for us. It actually allows us to see incredible detail of things that are really small.
You said the transmission, you can see the interior, and the scanning is the exterior?
Yeah, so
Okay.
these slides here are going over that. So we got the transmission electron microscope, can see the interior. Really is about all you need to know about it. It goes through a couple ideas there of how we can utilize them. But mainly, the biggest thing that I would ever ask you on any sort of exam is going to be interior for transmission, exterior for scanning. Here, like I said, scanning, they're much better, in terms of giving us 3-dimensional understanding of what the actual structures of different things are. So in this case, this is a bunch of bacteria. So we can actually see the individual bacteria. I can tell you right now they're bacilli because they're rod-shaped. And so we're going to see that as we go through in the next couple slides.
Since it has to be in a vacuum, does that mean like the bacteria?
Yeah, they're dead. Her question was, is electron microscopes a way to look at live, or are they all dead? They're dead. There's no way we can actually do electron microscopy on something that's alive. All right, the last one here, atomic force microscopes. These are the ones I was saying that we can actually look at the atomic structure of a molecule, and these are going to be very specific, very niched, as I pointed out in the very beginning of the talk. So what we're seeing here, this little white squiggly line, that's actually DNA. And then we can see those bright white spots on our enzymes, probably doing transcription or something of different genes. So that might be a polymerase on there. But we can see the individual enzymes attached to it. So that shows you how low we can go in terms of seeing details of different things. So most of what we do in microbiology, most of what we do in a hospital is all going to be fluorescent microscopy. Research, you might have confocals available. Atomic force is going to be very specific labs. Scanning electrons are going to be, and transmission electrons are going to be mainly in virology labs if they're doing research. In New Orleans, I think we only have 2 electron microscopes. Tulane and LSU Health Science Centers have 1 each. After that, there's really just no need for them. They're just so specific in terms of what they're used for.
You said the fluorescent ones are most common in hospitals?
Mm-hmm. So now we talk about staining. So how we actually stain things. What we got to refer to is there's lots of different kinds of stains, and there's different nomenclature we use for them. So we have basic and acidic dyes. So the basic dyes are going to be attracted to negative charges. Why is that important? They're the most common. What is negative on a cell? The phospholipid bilayer has a negative charge. So the outside of the phospholipid bilayer has actually got a negative charge to it. So these positive charged basic dyes will attach, right? Opposites attract. So positive and negative attract to each other. And so they're really good dye at staining the exterior of a cell. Dyes, and we're talking about pH there, right? Basic versus acidic. We're talking about the pH of the actual dye. Acidic dyes have a negative charge, and so the cells are going to repel it, and this allows us to see the outside, essentially. It stains the outside and allows sometimes the cell to pop. So most of the dyes that you're going to see, most of the dyes that you use are going to be basic dyes. They're going to be ones that are just looking at the outer surface. So this slide here is really good at going through all the different stains that we're going to look at as we go forward. I'm not going to waste any time on it because we're going to go over each of these on the slides that are coming up. But this is a good kind of review slide that you mark in your PowerPoints. All right, so the very first and the most common staining technique, one of the very first ones you learn as a student is the Graham stain. The Graham stain is named after Dr. Christian Graham that we'll see on the next slide. So it's not named after the metric unit of mass, so don't think it's Graham's weight or something. It's actually the person's last name. It's two groups. You're either Gram positive or you're Gram negative. And this is a huge tell for us in terms of differentiating what type of bacteria we might be looking at. Remember, all these stains that we're looking at from this point forward are all for bacteria, okay? They're not for viruses. They're not for any fungal, any eukaryotes. It's just bacteria we're talking about right now. And so Gram positive, Gram negative, it reflects the difference in the actual cell wall structure. So we haven't gotten into the basics of the bacteria except for there at the back end of chapter 1. We know that bacteria have a phospholipid bilayer, but on the outside of the phospholipid bilayer is the cell wall. And the cell wall is either going to be thick or it's going to be multifaceted, multiple stages to it. The Gram positives have a thick cell wall, and the Gram negatives are the ones that have multiple pieces to them, as we'll see as we move forward. So the way that the Gram stain works is there are 4 major steps to it. Step 1 is a stain called crystal violet, which is a purple color. That purple color stains everything on the slide. So if you have a mixed slide of bacteria, so you might have some Gram positive and Gram negatives on that slide, as we see here. That very first staining technique, the crystal violet, is going to make everything purple. Then we're going to come back and we're going to hit it with iodine, which helps kind of seal in the crystal violet. And then if it's Gram negative, when we hit it with the alcohol, which is just ethanol, when we hit the alcohol, it's going to actually knock out that purple color if it has the thin Gram negative cell walls. The ones that are super thick, the Gram positives, they stay purple. They're thick, the stain's stuck inside. Then we come back with what's called a counter stain. That's saffron. Saffron's got a pink color to it, and it's going to stain everything that got decolorized this pinkish color. Now the ones that are purple still take up the pink, but purple's darker than pink, so you don't see it. You're only going to see the pink in the ones that are Gram negative. So when we look at the difference between them, we can automatically tell, even if we don't know what bacteria we're working with, we can tell if they are Gram positive or Gram negative based on the color that they stain after a Gram stain. Purple are going to be the Gram positives, pink are going to be the Gram negatives. So Hans Christian Graham is the person that developed this. He was working in a morgue in Berlin, and what they were trying to do was trying to figure out a stain for tuberculosis. Tuberculosis has got a unique cell wall, as we'll see here in the next couple slides. It actually requires a completely different staining technique. And so at the time, he was like an understudy to the coroner, and he was charged as part of his job to come up with a way to stain Mycobacterium tuberculosis. And he wasn't able to do it, but with what he was coming up with, but he came up with this brilliant Gram positive, Gram negative, which helps us in differentiating what somebody might be sick with. So when we were utilizing Gram stains almost exclusively in the hospitals and whatnot, it was a big game changer because different antibiotics work differently, whether you're gram-positive or gram-negative. So if we can tell right away what somebody's sick with, it's even more important for us to be able to get them the right antibiotic. And so we'll talk about that more as we get into chapter 20 and the antibiotics that we use. So as I said, most of the bacteria we deal with are either going to be gram-positive or gram-negative, and that's based off the peptide glycan that's found in the cell wall. However, there's a couple different oddballs out there, and the two biggest ones are tuberculi and leprae, the leprosy and tuberculosis. They fall underneath the genus Mycobacterium. So see that myco, M-Y-C-O? That stands for mycolic acid. Mycolic acid is what these cells are made up of instead of peptide glycan. They don't have any peptide glycan in their cell wall. Instead, they're made up of this mycolic acid. It's a very waxy, fatty acid, so it repels the stain. It's really hard to stain them. So we actually have to use a little bit harsher methods to actually get these to stain. And so the name of the stain is acid fast, and you're either acid fast positive. If you're acid fast positive, you're probably a mycobacterium. If you're acid fast negative, then you're probably going to stain better with the gram stain. So one of the things that we do in the microbiology lab, I know most of you are nursing majors, you're not required to take our microbiology lab, but for those who might be taking it, one of the things you do is an unknown at the end of the semester. They give you an unknown sample of bacteria, and your job is to go through all these different stains to try to ultimately figure out what you actually have. And so the very first thing you would do would be a gram stain. So if the gram stain comes back inconclusive, your next step would be an acid fast, right? Because you're saying they're going, well, if it didn't go gram positive, gram negative, well, maybe it's a mycobacterium. And so you got to think about these stains as being sequential in the way that we would actually utilize them to ultimately determine what bacteria we're working with. Because at the end of the day, there's no way you can tell what that is just by looking at it, right? They're all going to be so small. They don't have any real true differential markers on the outside that we can just see with basic staining techniques. So we have to use some of these different stains that help us understand them. So the next one is the capsule stain. So on top of having the cell wall, some bacteria will actually have a capsule going around the outside too. So we have a bacteria. We have the phospholipid bilayer, right? So that's every cell has a phospholipid bilayer. That's what you've learned since freshman biology. They're all going to have a cell wall going around them. The cell wall is either going to be gram positive or gram negative based on how thick it is. So this will be the cell wall. And then some bacteria have the capability of putting a capsule around the outside. Not all, right? So again, this is helping us differentiate what we might have. The capsule acts almost like a camouflage against the immune system. So it's an adaptation that some bacteria have developed that helps them hide from the immune system. The immune system is used to looking for peptide glycan or another molecule we're going to talk about in this chapter called LPS. And so that capsule basically puts a cover over those, and now it gives the advantage to the bacteria, at least in the short term. And so one of the things I always like to talk about, is I like to play chess, but some of you all may not know chess that well, so think about whatever strategy game you might like. The immune system versus an infection is like a strategy game. For every single advantage that the bacteria has, the immune system is going to figure it out and has a counter to it. And then sometimes the bacteria has another counter to that. So it's a back and forth all the time. So the capsule is one of those strategies that some bacteria have to allow them to evade the immune system, at least for a short bit. And so we actually can stain for those, but we have to use one of those counter stains because the capsule is designed in such a way that it doesn't stain very well. So we have to use those negative stains, right? We have to use those acidic stains that we talked about that stains the background instead of actual the structure we're looking at. So what we see here is the capsule stain is staining the background, but here we can actually now stain the background. We can see the two bacteria and we see this clearing around them. That's telltale sign of a capsule. So at that point now we can say, okay, this bacteria that we're staining has a capsule around because we're staining the background. We get this big zone of clearance more or less around the outside of it. All right, next one, endospores. So we talked about this in chapter 1, right? How was it that, they had so much difficulty, fighting off the idea of spontaneous generation? Well, it was because some of the bacilli and the clostridiums can make those endospores, those really hard shells that go around them. Well, they're also something we want to be able to stain. So if they're in their capsule, if they're in, not their capsule, but they're in their endospore, we can't stain them. They're going to resist the gram stain because it's such a nice hard shell that it becomes, the stains that we use won't work very well. So what we have to do is we actually heat them up and force the stain to go inside. And in this case, we're going to be using something called malachite green. It's just another stain that's out there. And it's going to actually force itself inside the endospore. And then we can counter stain it. And then we can see the spores. Again, I'm not going to ask you any questions on how each of them work except for the gram stain. Make sure you know the gram stain like the back of your hand. The rest of them know what they're used for, not how we actually do them. Because most of you aren't going to ever be a technician where you're all be doing those things unless some of your med techs. Here, the last one that we have is a flagella stain. So obviously some bacteria have a flagella. Again, not all have them. So the ones that do, we have a stain that, will actually stain for the flagella, and we can actually see the flagella or the tails that they use for motility. And so each of these helps us be able to determine what somebody may be infected with or what type of bacteria that we're dealing with, if it's like a research unknown. So we move over to fluorescent dyes and tags. I already talked about how it's done. Again, it may not make a lot of sense to you past the surface level understanding until we actually do chapter 14 and 15 where we talk about antibodies more. But just keep in mind that these are designed or engineered, through biotechnology to take something that's natural, the antibody, and essentially attach a fluorescent marker on it. And that way we can utilize them to be able to visualize different cell types. So here you can see multiple colors. We can see that there's probably 2 different stains in here, staining 2 different types of bacteria. Here's one just using a singular color. Here's another one just using a singular color. But you can see that they come in a lot of different shades. I think the last time I checked, there's somewhere to the tune of 10 plus different colors that are out there available now. So that whole idea of utilizing fluorescent dyes and tags falls underneath the term immunofluorescence. Immunology for the antibodies, right? Because that's where we get them from, is the immune system. Fluorescence, because we're using them to fluorescently stain. So immunofluorescence is the name of the using the antibody to deliver that tag for the different proteins on the microbe. Okay, so those are the stains. So not a lot there. Just kind of keep in mind that you wouldn't necessarily use one by itself unless you were just training to use that one. You're utilizing them kind of in sequential order to try to come up with what your unknown is. And so each of those are going to give you a yes/no answer for the most part. Flagella, capsule, endospore, all those are yes/no. Is it there or is it not? And if it's not there, then we know that it's probably a different category. If it is there, then we know what category of microbe it falls under. Next part of chapter 3 is looking at the morphology. So morphology means what is its shape? What does it look like? This is without any stains on it per se. We're just talking about what does that bacteria actually shape look like. And so the 2 most common are coccus and rod. Coccus are going to be spherical, so think of them like a ball. The rod is going to be the ones that we looked at earlier, the bacilli. They're kind of more elongated. However, there are other shapes too. We got the Vibrio. Vibrio kind of have a comma shape to them. So the most common Vibrio is Vibrio cholera. Causes the waterborne illness cholera. Spirillum kind of has more of a wave shape to it. And the spirochetes have a corkscrew shape to them. So you think about like a wine corkscrew. And so the most common spirochete out there is Lyme disease. And what makes it so difficult to cure is because of its shape, that the spiral shape is it spirals right into the muscle tissue, and it makes it really difficult to get a drug delivered to that really tight, dense muscle tissue. And so there are several other examples of each of them, but the most common are going to be the cocci and the rods, but we will talk about some of the other shapes as we go through the different bacteria. There are also some that are pleomorphic. So some of the more common diseases that we'll talk about, we'll say that they're pleomorphic. Pleomorphic means that they have all kinds of different shapes. What is it that allows them to keep their shape? Their cell wall, right? Their cell wall is what's giving them their shape. So if they're pleomorphic, they may not have a very strong cell wall. They may have more looser type cell wall to them. And so they don't really conform to any specific shape per se. Most prokaryotes, so now we're talking about grouping them together. So here we're going to talk about how they divide. Remember, all bacteria divide by binary fission. That just means they're making a clone of themselves. They don't need a partner. There's no sexual intercourse involved for bacteria. They just basically go through the process of cell cycle and they split in half. And they made an identical copy of themselves because there was no exchange of DNA information. The DNA is going to be exactly the same. So it's an advantage here to be binary fission. Is the advantage is that you're able to increase your numbers very fast. Most bacteria, when they're in their optimal conditions, can replicate every 20 to 30 minutes. So you're going through a lot of generations and building up numbers really quickly. The downside is you're all exactly the same, right? So it takes a single drug to knock them all out. So when we look at how they actually divide, all bacteria divide by binary fission, but some of them divide in ways that actually give them different shapes. So we can actually start looking at those and looking at how they group together. So Neisseria gonorrhoeae, causes gonorrhea, is a diplococcus. Diplo, di, meaning two, right? So in this case, when they always divide in twos. So when we look underneath the microscope, that's probably one of the easiest ones. There's not a lot of infectious diseases that are diplococcus, per se, that we're going to cover in this class. So if you look at a microscope and you see two cocci in there, then you would say, oh, well, that might be Neisseria. Streptococcus, they make these long chains. So they start making chains. So when we something like bacteria that you've heard of, that's Streptococcus, you're causing strep throat. Right there, its name tells you. Strep tells you it's long chains. Coccus tells you that they're spherical. Yersinia, they divide in like these three-dimensional packets. I don't know of any particular bacteria that we're going to cover in here that are divided like that. But the next one, definitely Staphylococcus. So Staphylococcus aureus, very important one for medical purposes. MRSA, Versa, you heard of methicillin resistant. They make these grape-like. So think about going to the store and buying grapes. And each of the individual grapes would be an individual bacteria. So they kind of cluster up. And so that cluster gives it its name Staphylococcus. So this is how they get their names. Their names are based off of their morphology a lot of times. Some of them have multicellular associations. So in this case, all we're talking about is how they actually work together as a team. So even though they might be binary fission, some of them, when they swarm together, there's a bunch of them together, they have what's called quorum sensing. Quorum sensing just means it's their way of talking to each other. And when they recognize that that there's a large enough mass of them, then they'll start releasing enzymes or other organic materials that help them digest food around them for the whole. So a lot of bacteria will work together in teams, essentially, to get things taken care of. Especially when nutrients are limiting or water might be limited, they're going to conjugate. They're going to come together and try to, survive as a whole instead of as an individual. So now we're tearing apart the prokaryotes. So we're switching gears again. So we talked about the microscope, we talked about the stains, and we talked about the morphology. Now we're actually breaking down that bacteria sort of much better than my silly pictures over here on the whiteboard. So this should look pretty similar to you, right? You've probably seen pictures of prokaryote before. Got the nucleoid in the middle. That's going to be the area where the DNA is at. Again, it's not inside a nucleus. It's just kind of out there in the cytoplasm. There's really no specific spot. We just talk about the area where the DNA is located as a nucleoid. Of course, we got to have the ribosomes. Ribosomes are important for taking the RNA and translating it into protein. So we have all the information that we need in a eukaryote. We have it in a prokaryote too. We got DNA. It's able to transcribe into RNA. It's able to translate into the protein in the ribosomes. But everything else is going to be kind of different. We have the cell wall. We have the capsules. We have a flagella. So this bacterium here has everything, right? But remember, not all of them do. Some of them won't have a capsule. Some of them won't have that flagella. And it also has some here we haven't seen yet. The pilus. Pilus is singular. The more common word that we use is pili, P-I-L-I. So the pili, I always kind of think of as like Velcro. So the pili are going to be used for attachment. So if a bacteria, if you eat something that's contaminated with bacteria, it can't make you sick unless it attaches to your epithelial cells or actually attaches to some other cell type. Otherwise, it's just going to get processed right through, right? So it has to have a way to attach, to establish itself before it can even start an infection. So usually, the attachment is established with the pili around the outside. So this is a good slide to look at. Again, it's one of those tables I like to throw in if it helps you to be able to kind of have just one slide with a lot of information on it that's important. That's on there. But as we go forward, we'll see some of the things that are on that table. Okay. So most of this you've seen before. This should all be freshman biology throwback. The phospholipid bilayer. What is it? Obviously, it is the phospholipids. You got the hydrophilic heads on the outside of the cell as well as the inside, and the hydrophobic tails are facing each other. So that's why it's called a phospholipid bilayer. The fluid mosaic model basically was discovered in 1970s, and what it tells us is that all the proteins and different channels and whatnot that are found within the phospholipid bilayer, they're floating around. They're not stuck in place. They're actually able to float around to be able to get to where they need to go. But at the end of the day, there's no just opening. If you pop the phospholipid bilayer, the cell would die. But luckily, it can move around. Things can move around in there, and it doesn't actually cause any damage. Here again, this is all back to 141 stuff. Just talking about how does different molecules get in and out of the bacteria, out of that phospholipid bilayer. Simple diffusion. Remember, everything always moves from high concentration to low because it wants to be equal on both sides. So that's just simple diffusion. Osmosis is referring to water specifically and how it actually moves back and forth. So when we talk about osmosis, we're talking about the diffusion of water. So when we refer to different levels of osmosis, they fall under hyper, iso, or hypotonic, right? So hopefully those terms are sounding familiar to you. Hypertonic refers to the water basically being pulled out of the cell. It causes the cell to shrink. Isotonic means that water is moving in both directions the same, and so the isotonic is usually what human cells like. They like to be isotonic. Hypotonic means more water's flowing in than flowing out, and so the cell starts to expand. Our blood cells, if they actually are hypotonic, they burst, and you can actually die from that. We have a couple cases every year of people overhydrating, which sounds odd, but you can overhydrate and actually kill yourself. All your cells end up going hypotonic. However, things like plant cells and some bacteria cells, they prefer to be hypotonic. They want to swell up. They have that cell wall protecting them from bursting. And plants have a cell wall too, made of cellulose, right? So how is it that water comes into the ground, gets pulled up by the roots, and is able to get all the way up to the top of the tree? All those root cells are hypotonic, swelling up, and it forces the water up through the trunk all the way out into the extremities. A lot of bacteria like to be hypotonic too. So even though we prefer to be isotonic, our cells prefer to be isotonic, there are cases and ones that prefer to be hypo. I can't think of too many that like to be hyper, because hyper means that you're losing all your water. Electron transport chain, just a reminder of how ATP is made. The energy, is converted. We use glycolysis, pyruvate oxidation, Krebs cycle, all the way down to electron transport chain. Essentially, all the electrons get put on one side of the phospholipid bilayer and get put through the enzyme ATP synthase, and that ultimately produces ATP. Again, I'm not going to ask you any questions per se about things that you've already been tested over in other classes. This is just kind of a reminder to help you remember how these things work. So when we start talking about more complicated things in terms of how medicines and things get in and out of the cells, remember that there's a lot of this already in place by nature. Most of it that we do, most science, most medicine that we do, relies on something that's already naturally there, and we take advantage of it. So transport systems, you have carriers. Carriers can bring in one molecule at a time, whatever it might be that it's bringing in, but it has to be very incredibly specific, right? You know, we don't want to bring in everything. It's sort of like you don't want to leave your door open while you're not home, or even when you're home, you don't want to just leave your front door open, right? You don't want anybody randomly just coming in. So, you know, think about these carriers that kind of being the doors to the cell. You're going to allow one molecule in at a time in this case, and it's going to be incredibly specific for what it's actually looking for. But then we have some other ways, facilitated diffusion. If you're facilitating somebody, you're helping somebody. So it's just another word to say help. So in this case, it's a form of passive transport. When we use passive, that means there's no energy needed. And so this is going to be no ATP. It just happens naturally from high to low concentration diffusion, which is the second word, right? Facilitated diffusion. It's usually going to help bring in something else that's too big to come in on its own. So there's a lot of different examples of it, but just kind of think about facilitated diffusion is going to basically help bring in some other molecule that may not be the specific one that they're looking for. Active transport is when you're using energy. So anytime ATP is needed to bring something in, that's going to require energy, and these are going to be incredibly specific because our cells, including almost all cells in nature, they don't like wasting energy. And so they're not going to waste ATP to bring in stuff unless it's absolutely necessary and needed. And so usually if there's active transport involved, it is going to be something that the cells require. Group translocation, this is going to be moving large pieces, large amounts at one time. And so we'll see examples of these as we go forward. So here you got all four of them together. You got facilitated diffusion, active transport using ATP, and then group translocation, basically bringing in groups of things at one time. Just showing you all the different examples. So different medicines utilize these different types of transports. Different toxins that we'll talk about that bacteria are able to produce use these different types of transports. So they're all involved in the actual medicinal part of microbiology. This is just kind of the basic morphology of the bacteria itself. Okay, active movement out of the cell. So this is secretion. Secretion is going to be the way that we can actually use these channels to move things out. So the most common way that we do this would be extracellular enzymes. So enzymes that go to the outside. Those would be enzymes that might be necessary for chewing down a larger piece of food, a larger piece of energy source. Maybe too big for the bacteria to take on at once. They can push out those exterior, or they can secrete out those extracellular enzymes to help start breaking it down before it actually tries to bring in that energy source. They can also secrete out toxins, as we talked about already. There's lots of different bacteria as we go through the chapters that produce their own toxins, and they're going to secrete them out through these protein secretion systems. So moving on to cell wall, this is probably the most important part of all this. For this part of the chapter is looking at the cell wall to help you understand the difference between gram-positive and gram-negative. I find this incredibly important, so make sure that you're studying this. Don't go through those first few slides and go, I already know all this, and then skip over to cell wall. Make sure you're paying attention to how this one comes together. So cell wall, strong, rigid structure that prevents cell lysis, right? That's the whole point of it. One of the things that penicillin does is it actually breaks down the ability for the cell wall to form. Once the cell wall can't form, the bacteria just kind of breaks and falls apart and dies. And so cell wall, strong, rigid structure that prevents cell lysis. Architecture between two main types, gram-positive, gram-negative, based off that gram stain. And both of those, gram-positive and gram-negative, are going to have peptidoglycan in them. Peptidoglycan is the molecule that they're made up of. Again, only found in bacteria. We haven't changed anything yet. We're just still talking about bacteria. So what is peptidoglycan? Well, we can break it down even further. Peptidoglycan is made up of two molecules, N-acetylmuronic acid, or just abbreviated NAM or NAM, and N-acetylglucosamine, which is abbreviated NAG. And all it is going to be over and over again, NAG, NAM, NAG, NAM, and that makes peptidoglycan. They're held together by these tetrapeptides, so basically a string of 4 amino acids that act like little screws or nails holding them together, holding each of those sheets of peptidoglycan together, and that starts forming the cell wall. So when we look at gram-positive, I want you to make sure you're paying attention to the actual structure that you're seeing here. Make sure you recognize at this bottom, that's the phospholipid bilayer, right? Every single cell in nature has a phospholipid bilayer. We can see all the different proteins and carriers and channels that are kind of embedded in there. But then notice on top of that, as we draw, you get the cell wall that sits on top of that. This one here is gram-positive. Remember I told you the gram-positives have a thicker cell wall. So in this case, the thicker cell wall, we can see all the blue and purple dots. Those are the NAG and NAMs coming together making the peptidoglycan. We can see the tetrapeptide bonding there, holding them together. But then we have something else happening there, right? We got those little red lines coming down. Those are the teichoic acids. Those are what's actually anchoring the cell wall to the phospholipid bilayer. So see how it goes all the way through and actually attaches to the phospholipid bilayer? That would be a gram-positive. Gram-positives are pretty straightforward. They just have that peptidoglycan layer attaching it all together with the teichoic acids, holding it to the actual phospholipid bilayer. Gram-negatives, a little bit more complicated, but once you get through the image here, it shouldn't be too bad. You have two layers. You have an inner membrane and an outer membrane. So we have the inner membrane is your original phospholipid bilayer, right? So that would be the one that we always think about when we think about a cell. We have all the different proteins and stuff that are embedded, right? Just like the gram-positive. Then we have what's called the periplasm. It's this empty space between the inner membrane, and then there's another outer membrane of phospholipid bilayer. And so in between that is the periplasm, and that's where the peptidoglycan is located. So that's what makes it gram-negative. Remember I said that it has a thinner amount of peptidoglycan? It's just this really thin amount of peptidoglycan that's just found in the periplasm. Then you have the outer membrane, and then there's an incredibly important part that's attached to the top of that. The outer membrane is attached, it's called lipopolysaccharide, or LPS. Lipopolysaccharide is a what we call an endotoxin. So endo means it's attached to the bacteria. It doesn't get secreted. It actually stays attached to the bacteria. It is the only endotoxin that we are going to talk about in this this semester that's important medically. A lot of people end up with sepsis. It's because of LPS. It's because they're infected with some sort of gram-negative bacterium. So the LPS layer is the most important. Why is it so important? It's on the outside. That's the first thing the immune system is going to see. It's not going to see the outer membrane. It's not going to see the peptidoglycan. It's not going to see any of this. First thing it's going to see is the LPS attached to the outside. So that's one of the big parts of gram-negative. It has three parts to itself. So this little corner, this little box up here on the top right is referring to what LPS looks like when we take it apart. It has that lipid. Well, remember it's attached as part of this outer membrane, so that's the phospholipid. That's a modified lipid that allows it to be part of that phospholipid bilayer. It's going to have a core structure here in the middle. It's just kind of the stuff holding it all together. The most important part is the very outer tips. We call that the O antigen. And so the O antigen there's lots of different O antigens out there. And antigen, we'll talk about when we get into the immune system chapters, but antigen refers to antibody generator. So anti is the antibody, generator. That's where antigen comes from. And antigen is anything that causes the immune system to have a response and start producing antibodies. So in this case, that's where the name antigen comes from since the first time we've seen it. So all this basically refers to what I already talked about. You have lipopolysaccharide, LPS, incredibly medically important. LPS is the only endotoxin that we'll be referring to. All the other toxins we talk about in this chapter, in this class, are going to be what we call exotoxins. Exo meaning that they're secreted through one of those secretion systems we were just seeing on the slide before. Periplasmic space. Periplasm, just basically these slides are referring to what I already spoke about, so nothing new to see in these slides. Already talked about penicillin interferes with peptidoglycan synthesis. We'll talk about that more in chapter 20, I believe, when we get into the different antibodies. We'll actually break it down, but just as like a starter, penicillin is our first antibiotic that we use, nationwide, essentially, or worldwide. There were a few before it that we'll talk about when we get to that chapter, but penicillin was the most common, most readily available, came on the scene in the early 1900s. Nobody knew how it worked at the time. It just worked, helped cure people. Like you said in the very first chapter, if you got sick in 1800s or earlier, you're pretty much dead. Your body, unless your body is able to fight it off, if you got some sort of bacterial infection, there really wasn't much hope for you. Penicillin came on the scene, helped change that. Well, we wanted to know how it worked. And so what we found out was it interferes with the bacteria's ability to make peptidoglycan. And so what that does is it prevents the cross-linking of those glycan chains. And so what that means is essentially, let's go back up. All these cross-linking chains, it basically keeps them from being able to form. And so the whole peptidoglycan layer can't form. The next bullet that was on that slide says that penicillin-type drugs are much more effective against a gram-positive. Why? Because the peptidoglycan is the first thing the antibiotic sees, right? And so it's going to be able to interfere with it or mess with it the best. If it was a gram-negative, peptidoglycan layer is clear down here inside. So somehow that drug has to get past LPS, past that outer membrane to get to the peptidoglycan. It's a lot more difficult for penicillin to work. So that's why I was referring, coming full circle as we're running out of the last few minutes here, coming full circle to the idea that it's important medically to know what somebody's infected with and doing that gram stain right away. Are you gram positive or gram negative? If you're gram positive infected, we may be able to get away with just giving you penicillin. If you're gram negative infected, we may have to go with a stronger medication, stronger drug to understand it. Nowadays, we got better diagnostic tools. We're not as reliant on the gram stain as we used to be. However, even some of the newest diagnostic tools still rely on the ideology behind the gram stain. All right, so keep moving here. Lysozyme, so this is something we'll talk about in the immune system chapter also. Lysozyme is a natural enzyme found in our tears as well as our saliva. And what it does is, our tears, why do you start tearing up? You get something in your eye. It's an immediate immunological response to prevent any sort of bacteria from, infecting your eye. It's in our saliva because we eat a lot of, we eat all the time, right? And no matter how clean you think your food is, it's not as clean as you think it is. And so having the lysozyme, this is just a natural generic enzyme that's found in our tears and saliva and other bodily fluids that also messes with that peptidoglycan, the ability of peptidoglycan to form. So some bacteria lack a cell wall. So things like mycoplasma, right? We talked about needing an acid-fast stain for them. The acid-fast stain helps us see that mycolic acid, but what is it that mycoplasma doesn't have? It doesn't have peptidoglycan, right? So that's why we have to use the acid-fast. There's nothing for the gram stain to work on. There's no gram-positive, gram-negative for these guys. So if you're infected with something like tuberculosis or leprosy, penicillin is absolutely useless, right? Because penicillin works on breaking down the peptidoglycan layer. If you don't have it, it's basically you're just drinking some water and thinking it's going to cure you. So again, knowing what you're infected with and understanding what the actual structure of those bacteria are helps you understand how the medicines work too. So here, members of Archaea, my meme's outdated. That was when Charlie Sheen was going nuts. In this case, Archaea, one of the things I want you to know about this is something I already talked about last week. In the Archaea, there's amazing microbes, but there are 0 human pathogens. Why? Because the Archaea include microbes that live in extreme environments. In order for a microbe to cause disease in us, they have to live in the same areas that we do. They have to be exposed to the same things that we're exposed to. I don't think anybody can go survive in a volcanic vent that I know of as a human anyways, but there are Archaea that do. We've actually been able to pull microbes out of the Antarctic going down like 30, 40, 50 feet into the ice and have pulled out microbes that are actually still alive. Like they're alive down there. however they're working, I don't know, but they're all part of the archaea domain. And so because of where they exist and because humans aren't found there too, there are 0 human pathogens in the archaea domain. These slides just kind of talk about some of the differences, but there's really not a whole lot important there, except for there's no peptidoglycan found in the archaea domain either. So capsules and slime layers, we already talked about capsules briefly. Capsules are going to be very distinct. They're like that camouflage that goes around, but there are some other bacterium out there that forms what's called a slime layer. They're more diffuse. They don't have that distinct look to them. And so, again, they help us understand what type of bacteria we're dealing with, whether they make a slime layer or a capsule. The ones that make these slime layers or capsules, some of them can actually also make what's called a biofilm. The term that's important on this slide is biofilm. Biofilms are what we are currently fighting, and there have been some amazing strides in trying to combat biofilms. What occurs in a biofilm is, think about something that's like plastic or something like an artificial hip or an artificial knee. One of the things that we got to make sure that's incredibly important is that it's sterilized, that there's absolutely no microbes on it before it goes in. Catheters, same way. So what occurs is that, if you have, let's just use a catheter idea, so it's usually like a tube, right? A couple bacteria get in there. What happens is that they actually can attach to the plastic. And what they'll do is they'll start forming a biofilm over the top. And they start building their numbers underneath there. What occurs sometimes is that, these pieces will break off. And if it's going the wrong way, it goes inside the body, it can actually cause major infections. It can cause major problems. So you see, it can be not only catheters, but artificial knees, artificial hips, all problematic for biofilm formation because we don't need that. Most of the time, if you're getting any of those type of medical procedures, you're usually a little bit advanced age, right? Your immune system's not functioning all that well. And so the last thing that grandma or grandpa needs to go through is getting a second hip or getting a replacement or something like this because one ended up being infected. Nice thing is now that we understand this, we are getting much better at making plastics and things that the bacteria cannot attach to. So some of the new generation medical devices are basically biofilm resistant. But one that you are all familiar with is waking up in the morning and having that filmy taste to your mouth and feel that film on your teeth. That's actually a biofilm formed by the bacteria Streptococcus mutans. And so we'll talk about that in the digestive disease chapter. One of the very first diseases we talk about in that chapter is cavities. And so that actual film is those bacteria forming those dental plaques. And ultimately, if they allow it to continue and you don't brush it off, you end up with cavities, right? So we'll stop there for today. We're out of time. So hopefully you all like this room. We'll keep meeting here unless you hear otherwise. We might even be able to take our exams in here because we can use the computers. And you can take the exam directly on the computer, just like my online classes. So I'll work on that in the next couple of weeks. Again, if you want to take a paper version, I can always bring paper exams in too. This depends on how it goes. So we'll see you all on Thursday.